The overall goal of this optical imaging technique is to provide a means for rapid screening of novel bacteria-specific optical probes which may have clinical tractability for imaging infection within human lung.
This method can help answer key questions in the molecular imaging field, such as whether novel optical compounds are specific to their target and whether they are detectable.
The main advantage of this technique is that it's translatable. Selecting promising imaging probes using equipment that's available in the clinic provides confidence that the probes will work in that environment.
Immediately before imaging, remove the human lung tissue sample from the freezer on dry ice. At room temperature, allow the tissue to thaw slightly so that it can be sliced with a scalpel into 1 by 4 mm sections. Using forceps, place the sliced human lung tissue into wells of a 96-well clear flat-bottom tissue culture plate. Return any unused human lung tissue immediately to the storage container and place on dry ice for transport back to the -80°C freezer. Next, add 100 microliters of PBS to each lung tissue sample with a pipette to wash the blood from the sample. Use the pipette tip to ensure all the tissue is covered in the PBS and not stuck to the walls of the well. Remove and discard the PBS. Then, pipette 100 microliters of unstained calcine AM labeled or test probe labeled bacteria to each well containing lung tissue. Also, set up controls with lung tissue and 100 microliters of PBS. First, clean the fibered confocal fluorescence microscopy imaging fiber connector.